Thank you for having the possibility to present in ZMT Reef talks this study case that involved different key actors in the study area during the time I was doing my doctoral research, and that remains in progress as a multidisciplinary effort for the conservation of coral reefs in Dominican Republic. And this work actually conducted when I was PhD student at the Research Center and Advanced Studies of Politécnico Nacional de Mexico, better known as SIMBE-STAF, under the advising of Dr. Ernesto Arias and with the logistical and aquatic operations support of Fundemar in the Dominican Republic. This is the story behind this study and the presentation of our most recent work by myself, Aaron Muñiz and Ernesto Arias from SIMBE-STAF, Camilo Cortez and Megan Morikawa from Wave of Change of the Iberostar Group, and Rita Sellares from Fundemar. Thinking that maybe this presentation can be seen by different kind of audience, I always like to start by mentioning the complexity of the biology and ecology of corals, because they are animals that establish close relationships with their symbionts, dinoflagellates, and different microorganisms that apparently play a very important role in the development and response of their immune system, and that in the scientific literature is increasingly mentioned under the concept of holobiont. We know that every change in the environment in which they develop has repercussions not only in the physiology of the colonial animal and its polyps, but also in the composition of their bacterial communities, their symbionts, and the microorganisms that are associated in their mucus and sediments. So these organisms represent a complex interactive system with the potential to extend the physiological capabilities of the coral. No? So I like to mentioning the complexity of the effects of global change on coral reefs too. I like to use this diagram described by Preslowski et al. 2008, where we can see the relationship between the different processes and potential stressors and the possible responses at the level of organisms and communities. For example, global warming process could be related with salinity and temperature, and these potential stressors could be related with organismal responses like reproductive timing and larval success, for example. And this could continue to the response at community level, like dispersal and recruitment, community shifts, or habitat loss. One of the most affected genus in the Caribbean region has been genus Acropora. It presented a decrease of about 97% of its population since the 80s. Mainly the authors who have reported this way attribute this decrease to the white band disease, hurricanes, storms, increasing the average sea level, and pollution. The two representative species in the Caribbean, Acropora palmata and Acropora cervicornis, are listed by the IUCN as critically endangered. These two species have been perfect candidates to be used in restoration programs in the region because they provide very important ecological functions such as coastal protection, habitat provision, and refuge from thousands of reef species. In addition, they have lost between 80% to 90% of their populations and have failed to recover in the last 10 years or 3 generations, and also have high growth rates compared to other mass growth type species. In the Dominican Republic, Acropora cervicornis is the species most used in restoration projects that maintain them in nurseries and outplanting areas. Until 2015, it was estimated that its restoration was successful in terms of survival in nurseries and outplanting sites. However, the effect of restoration on coral reefs was unknown. We didn't know if the nursery colonies were spawning. We also didn't know what was the genetic diversity of the populations in the mother nursery or main nursery, nor what was happening on the reefs where we outplanted colonies of Acropora cervicornis. So the pilot project of this program started in 2011 with a nursery formed by 4 structures, each supporting around 30 fragments. Most Acropora cervicornis fragments came from one of the Ecological Punta Cana Foundation nurseries with multiple genotypes collected in different zones of the island, such as Punta Rusia, Samaná, Bávaro, Punta Cana, and La Caleta. Other fragments were collected occasionally in the area of Bayaibe. And in 2017, there were a total of 8 nurseries and 6 outplanted sites, all located within the Arrecifes del Sureste Marine Sanctuary, declared MPA in 2009. Despite being MPAs, it attracts 2,000 to 2,500 tourists each day, with an annual average of 600,000 visitors, representing $250 million per year. Consequently, the impact in the coastal marine ecosystem is important. Until 2015, the only technique used to make restoration was to propagate it by fragmentation, by asexual reproduction. That is why we were interested in starting to use different propagation techniques, among them sexual reproduction through embryo larval and recruit rearing. This was the first time that the spawning of Acropora cervicornis was recorded in a nursery in the Dominican Republic, and it was the first study of assisted sexual reproduction carried out for this species in the DR, and was the starting point to continue and optimize breeding techniques for the sexual reproduction of embryos, larvae, and recruits of Acropora cervicornis in the island. We did all the training work for representatives of Ministry of Environment of the Dominican Republic, park ranger of the national park, local fishermen, businessmen, diving instructors, international students, and of course, the Fundemar team. Simultaneously, we were working on assessing the restoration program and genotyping Fundemar's main nursery. The 2 main objectives of this study were to assess the coral restoration program over time, analyzing the results within the context of the regional restoration benchmark for Acropora cervicornis proposed by Schopmeyer et al. 2017. And in the second place, to determine the genetic diversity of Acropora cervicornis colonies in the main nursery for use in the future regional restoration efforts. The restoration program examined in this case study has provided a number of benefits for the local ecosystem and economy, such as maintaining genetic diversity in nurseries with 32 available genotypes, creating outplanting sites that have contributed to the rapid creation of fish and invertebrate habitats that would otherwise take decades to form, providing a sustainable source of corals for experimental research, like in my case, providing unique volunteer and employment opportunities for local communities interested in participating in restoration progress. We also reported that mortality in outplanted sites was associated with the presence of predators, mainly fireworms. Therefore, consistent maintenance and cleaning of outplanted sites is equally important than maintenance in the nurseries. At the same time, Dr. Camilo Cortés would hear these studies where we recorded 14 new records of fishes present in coral reef of this region. Also, we highlight the importance of having a baseline of ecological indicators, as well as providing an overview of trends in the reefs of the Southeastern Dominican Republic. For example, in this paper, we demonstrate how using different active and passive restoration techniques, as well as involving key stakeholders from government, academia, and local communities can improve the ecosystem functions of these Bayaibe reefs over time. These three studies are being used by representatives of the Ministry of Environment and Natural Resources of the DR to prepare a baseline report on the status of the fish species that will be closed for 2 years by official decree. Finally, our most recent work, in which we describe why it is important to perform analyses that include different variables to help us evaluate whether active restoration actions are recovering the ecological functions on the reef. In the Caribbean, the endangered staghorn coral Acropora cervicornis has been studied for restoration for over 2 decades, with most studies focusing on evaluating simple metrics of success, such as colony growth and survivorship in both nurseries and outplanting sites. However, for reef restoration to aid in the recovery of ecological function in outplanting sites, there is a need to measure the functional ecology of the impact of outplanting. Here, we present and identify positive ecological processes and ecological functions, such as increased fish biomass, coral cover, and increased in structural complexity relative to active reef restoration. We monitored the percentage of benthic coral cover and fish biomass alongside active reef restoration over the period of 12 months in 4 zones. Subsequently, we developed multidimensional analysis in conjunctions with GLM and linear models. Species compositions showed a marked differentiation among the study sites considered. This variation was significant for benthic coverage, as well as in coral and fish species. Similarly, the variation in species composition over time was significant in the benthic groups, corals and fish species. The difference in species composition is greater only between zones or only between dates, but which each site between dates there was not significant, was not statistically significant. Regarding to differentiation in fish species composition between zones, 6 of the 41 species registered was significant. In fish species, the only significant temporal variation was the species Scarus iseri, which presented a considerable increase, especially in the outplanted sites. Analysis of the regression models showed clear ecological benefits due to transplantation, mainly expressed in increased coral cover and increased structural complexity, evidenced in the Reef Functional Index. The increasing habitat complexity and coral cover also reflected in an increase in total biomass for all fish species sampled, especially for Scarus iseri. To our knowledge, this is the first study to explore the restoration of ecological functions at Acropora cervicornis outplanted sites in the Dominican Republic by including approaches using multiple variables to describe the correlations between the habitat studied. And the assessment conducted here indicates that species restoration induce habitat rehabilitation, increasing coral cover, reef functional index, and biomass in this particular case study of Scarus iseri. Damage from storms or hurricanes would be even greater if no active restoration efforts were undertaken. Continued active restoration efforts will improve the ecological health of coral reefs. Our results suggest too that Acropora cervicornis may be an optimal habitat that can facilitate the reintroduction of Scarus iseri, and it is likely that the fish species is a good indicator of improved fish recruitment condition as a function of habitat improvement. Considering that this fish is associated with highly complex reefs, it is probably that increasing reef functional index will be reflected in increased Scarus iseri due to increased habitat availability. Among all these management strategies, the private sector has played a significant role. Iberostar Groups, Wave of Change is a pioneer movement in responsible tourism and strives to set the standard for the private sector's care of the coastal ecosystem that provide critical ecosystem services to its businesses. The private sector has been viewed as a potential candidate for providing investment and new solutions. We are working on standardizing restoration programs across multiple locations in the Caribbean where we operate, so we can best understand how tourism can catalyze solutions. We have three pillars of action. Circular economy, responsible consumption of seafood, and coastal health. We are already single-use plastic-free in all of our plus than 120 hotel operations. We want to continue working on being carbon neutral by 2030, waste-free, and providing 100% responsible sourced seafood by 2025, and of course improve the health of the ecosystems surrounding our properties. Through our network of restoration efforts, we are already contributing unique findings. For example, my colleague Macarena Blanco is conducting a study to determine the temperature at which 50% of bleaching response is observed of 6 common reef building species in the Caribbean, with the aim of restoring ecosystem services of coastal protection while remaining resilient to climate change. This work was developed in our own coral lab, built in one of our hotels in Bávaro Resort in the Dominican Republic. This work, as well as our work in Mexico, is also featured in the recently published manuscript by Elisa Bayraktarov et al. 2020, summarizing the efforts of reef restoration in Latin America more broadly. Building of the success we have been able to demonstrate in the Dominican Republic, we are working to replicating aspects of this work in Mexico, Jamaica, and soon in other destinations where we operate. We are also working to expand the base of research that help us to scale reef restoration in all our destinations. And this is all from my side. I want to thank you very much for your time. I thank Sonia for inviting me here. I'd like to thank to all co-authors, colleagues, institutions, and people who participate and continue working for our coral reefs in the Caribbean region.